So our next biological molecule is the nucleic acids, and these are what we would refer to as the genetic material of the cell. So as we'll talk, DNA is one type of nucleic acid, and that is what serves as the genetic instructions. That's what's inherited. And in the very first cell of a new organism, this genetic material, this DNA, is what instructs the development of this organism and how the cells will differentiate and become different types of cells. So this is an example over here to the right of the monomer of nucleic acids. The monomer of a nucleic acid is called a nucleotide, and I mentioned to you that this is what we refer to as the genetic material of living cells. DNA is one type of nucleic acids, and the other type is RNA. Now, I mentioned to you the DNA is the inherited material. This is essentially what instructs the development of an organism. It instructs the happenings within a cell. DNA exists as a double-stranded molecule. We'll look at the structure a little closer in a minute. The RNA is actually sort of an intermediary molecule, and later in the semester we'll learn details for this, but the DNA in a cell like ours, in a eukaryotic cell, doesn't ever leave the nucleus. But proteins are produced outside of the nucleus. So the RNA is sort of this messenger, this intermediary that carries the instructions that the DNA has out so that proteins can be made based on this particular set of instructions. Now, RNA is different from DNA in that it usually exists as a single-stranded molecule. But they both are made up of the same type of monomer called a nucleotide. And the nucleotide has three components that we'll look at in its structure. So the first is right here is a sugar. So notice, remember, there's that ring structure that we saw before. Now this one looks a little bit different because the ring structure we saw before actually had a six-membered ring. This one only has a five-membered ring and then a carbon coming off it, but it's still a sugar. The other component, or one other component, is what we call a phosphate. That was one of those functional groups that we looked at the beginning of this lesson. And the third component is what we call a nitrogenous base. Now, sometimes I'll just refer to it simply as a base. Nitrogenous meaning that it contains nitrogen, and you can see, in fact, that it does have multiple nitrogen atoms as part of this particular base. Now, for the monomer, for the nucleotide, there are actually 4 DNA monomers, okay, and there are 4 RNA monomers. And we'll talk about those in a second, but just for a minute, several of them are the same, except, so all, DNA and RNA both have A, C, and G monomers, but DNA has a T monomer, whereas RNA has a U. So the way we can think about that is, when we talked about proteins, we said there were 20 different amino acids, so there's 20 different types of monomers. For DNA, there's only 4 different types of monomers, and for RNA, there are 4. So this part here, the part that says nitrogenous base, that's the part that's going to be different depending on whether we're looking at an A or a C or a G or a T monomer. Now, I mentioned to you A, C, G, and T. Now I have them spelled out for you. Adenine, guanine, cytosine, and thymine. Those are the 4 essentially DNA, the letters of the DNA alphabet. There are only 4 of them. And I mentioned to you for RNA all are the same except for one. Uracil replaces thymine. Now, I also told you earlier that DNA exists as a double-stranded molecule. What that means is there's essentially 2 separate molecules or strands of DNA that are held together. And this is a model representing what DNA looks like. So there is one strand, we would call it, if you follow this ribbon, okay, that ribbon is part of one strand of DNA. And I'm going to change colors here. And this strand here, or this ribbon here, is a part of another strand of DNA. Now, they're twisted, but if you imagine that they look like a ladder before it was twisted and then the ladder was twisted, basically it's the edges, right? The ends of the ladder that those ribbons are representing. Now, those two ribbons are actually held together by what's going on in the middle of that molecule. So we see that there are some structures here that are sticking out, right, from the ribbons. And those structures that are sticking out are actually those bases or those nitrogenous bases that we looked at. And so what's happening is those bases are coming together and they're forming a pair. So every time there is an adenine base, let's say if this were an adenine base here, it's pairing with a thymine base on the other strand of DNA. So that would make this one here a thymine. Also, guanine is always pairing with cytosine, and they do this. This is the only way that they pair unless there is some kind of a mutation or an error. So that pairing is actually what holds the two strands of DNA together. And it's something that we learned about in the last lesson called hydrogen bonding. And remember, hydrogen bonding is a much, much weaker bond than a covalent bond. But in DNA, all of these collective hydrogen bonds between all of the bases have enough energy in them to hold those two strands of DNA together. However, as we go through the semester, what you'll see is there are times when the DNA double-stranded molecule actually needs to be separated, maybe when cells are dividing and it needs to be copied, or when a particular gene needs to be turned into its protein product, then it needs to, the RNA copy needs to be produced. So that's why that hydrogen bonding is important. It holds them together, but it can be broken and those strands be separated when it's necessary. Now, we know that there's three components of a nucleotide. There's a phosphate, we looked at that, and there's a sugar, and then there's the base that's holding it together. Now, when we say the backbone of DNA, what we're talking about is this ribbon that I showed you. So that's what we mean by the backbone. So we know that it's the bases that are in the middle that are holding the two strands together. So this ribbon that we see is actually chemically made up of the phosphate part of the monomer and the sugar part of the monomer. Now, all of those bonds that are occurring are covalent bonds. Those are very strong chemical bonds. So the individual strands then are just held together by these nucleotide hydrogen bond bases that we mentioned. So let's take just a minute and look at this in a little bit simpler form and make sure that we understand. Let me change back to a color that's a little easier to see. So as I mentioned to you, this would be like the DNA molecule before it's sort of twisted into that helix. So we would consider that the sides of the ladder over, this side of the ladder and this side of the ladder, okay, those would be what we would consider the backbones. Okay, so this would be one strand of DNA, and this here would be another strand. Now the rungs in the middle, these are held together by the hydrogen bonds between the bases. So for example, if this structure is an adenine, then we know that this over here must be a thymine. If this is a cytosine, this must be a guanine. And it's the hydrogen bonds that are occurring between those. That's what's holding the two strands or the two sides of the ladder together. Now if we take a close look, what exactly do the sides of the ladder even look like? Okay, now the sides of the ladder are made up of the sugars and the phosphate for each of those monomers that we looked at. So obviously this is not going to be drawn with the full structure or even to scale, but in other words, what you can imagine is if there's a phosphate right here, I'm going to represent it this way, then that would be bonded to the sugar molecule, And coming off of that sugar molecule then would be, right, one of the bases. So let's say this one is a guanine. Okay, then this would continue on and we would have another phosphate, and then we would have another sugar. And the phosphate sugar, phosphate sugar, phosphate sugar, that is what's making up the sides of the DNA strand or DNA ladder. Same thing would be happening on this side, except this would be going in the opposite direction. So if this side of the ladder is running in this direction, the DNA strand, then this one is what we call antiparallel. It's running in exactly the opposite direction. Now this has got a lot going on in this figure, but this is representing the differences and some details about DNA and RNA. So this ladder over here, this is the DNA ladder that's twisted. So we looked at it and we said, okay, the ribbon we know, or the sides of the ladder, are made up of that sugar, phosphate alternating. Then these rungs of the ladder are the hydrogen bonds that are occurring between the bases. And so what you can see here is the actual chemical structure of each of the nitrogenous bases, or the 4 possible bases in the DNA alphabet. Over here in the RNA, on the RNA side of the slide, you can see that RNA usually exists as a single-stranded molecule. So it's only got one of those sides or one of those ribbons. So it's not hydrogen bonding with anything else. And you can see that the uracil here is different chemically than the thymine that's present in DNA.